Hi everybody, welcome back to the Mutations series of lectures. In the last lecture, we finished by discussing some causes of point mutations. In this lecture, we will look at an entirely different group of mutations, insertions and deletions, or indels. Recall that a point mutation occurs when one nucleotide in one strand is substituted with another. This is why point mutations are formally referred to as single nucleotide substitutions. In point mutations, there is no net loss or gain of nucleotides. The nucleotide is simply altered or replaced with another. With further series of DNA replication, this newly synthesized, or rather newly replaced nucleotide will go on to be paired with a different nucleotide on the opposite strand, according to the rules of complementary base pairing. However, with point mutations, there will be no additional or subtracted nucleotides from any newly synthesized strands of DNA. In contrast, insertions and deletions involve the net gain or loss of nucleotides. Insertions occur when segments of double-stranded DNA are inserted somewhere that it had not been previously. The inserted segment of DNA may be as small as a single set of paired nucleotides, with one nucleotide added to each strand. Deletions occur when a segment of double-stranded DNA is removed from its normal position. Again, this deleted segment may be as small as a single set of paired nucleotides, with one nucleotide subtracted from each strand. Insertions and deletions are often categorized as falling within two different size categories: large and small. By convention, the length of an inserted or deleted segment that distinguishes an indel from being large or small is 400 base pairs. It is important to remember, however, that this is a line drawn in the sand. Any segment over 400 base pairs is considered to be large. Generally, these large insertions and deletions involve large-scale changes in the number and location of several genes on a chromosome. Some can be visualized microscopically with techniques such as fluorescent in situ hybridization, which we discussed in the Chapter 2 material. Because of this, large insertions and deletions often are categorized as chromosome mutations. Recall that chromosome mutations are mutations that entail changes in a portion of the structure of a chromosome. One exception to this generality, however, would be insertion and deletion mutations arising from the movement of transposable elements. Recall that from mutations 2, transposable elements are interspersed repetitive sequences, and that interspersed repetitive sequences are repetitive DNA segments located thousands of times interspersed throughout the genome. These interspersed repetitive sequences have the unique ability to jump from one area of the genome to another. Remember that this isn't characteristic of all repetitive interspersed sequences. It's only the repetitive interspersed sequences that are classified as transposable elements. These transposable elements can range in length from 1,000 to 40,000 base pairs. This puts them in a category of large insertions and deletions, but also scales their size to the level of individual genes. For example, often transposable elements will cause disruptive mutations when they insert themselves into the coding strands of individual genes. As a result of this, large mutations resulting from transposable elements are more appropriately described as gene mutations. Recall that gene mutations are mutations that result in changes in the DNA sequence of individual genes. All right, and here in the picture we can see the two different main categories of transposable elements. On the left you see a transposon, where literally that piece of the repetitive interspersed sequence is enzymatically cut out of the DNA molecule and then transplanted somewhere else. And then over to the right, you see a retro transposon, where that repetitive interspersed sequence is transcribed, all right, and then that mRNA transcript is reverse transcribed back into a repetitive interspersed sequence of DNA, and then that repetitive interspersed sequence of DNA is planted somewhere else in the genome, much like the transposon was. So in contrast, insertions and deletions falling under the category of chromosome mutations are caused by errors in crossing over. And remember, crossing over is just another way of saying the recombination that occurs during meiosis I. So as a result, large insertions and deletions are characterized as chromosome mutations, will always begin in the germline, and therefore these chromosome mutations that are insertions and deletions are always considered to be germline mutations. We will consider the mechanisms by which such large insertions and deletion mutations occur during our cytogenetics lecture. So again, over here on the left, you can see a review of the large insertions and deletions. All right, and now we're going to move on to small insertions and deletions. So the focus of this lecture is going to entail small insertions and deletions. Specifically, we will look at the nature of small insertions and deletions and discuss the circumstances in which they occur. As a brief introduction, small insertions and deletions involve segments of double-stranded DNA 1 to 400 base pairs in length. They are categorized as gene mutations as their impact is scaled at the level of individual genes. Again, recall that gene mutations are mutations that result in changes in the DNA sequence of individual genes. Small insertions and deletions are caused by errors in DNA replication. These mutations can either be germline mutations or somatic mutations. If these errors occur in the germline, they will be considered germline mutations. If these mutations originate in a somatic cell, they will be considered somatic mutations. As previously noted, small insertions and deletions are caused by errors in DNA replication. The first box within this series of animations demonstrates normal DNA replication. The blue line with the green bar in the center represents one of the strands of a separated DNA molecule. Keep in mind that this strand is a single strand of DNA. It is not double-stranded. Notice that this single strand of DNA is oriented in the 3' to 5' direction. Now we will demonstrate the synthesis of a new strand that is complementary to the parent strand. Notice that this newly synthesized strand is oriented in the 5' to 3' direction, and that its nucleotides are laid down and connected in the 5' to 3' direction. Recall that the enzyme that lays down the nucleotides of the newly synthesized strand is called DNA polymerase. And DNA polymerase does it in such a way so that these nucleotides are connected to one another and complementary to the nucleotides of the parent strand. To review DNA replication, please refer back to the Chapter 2 notes. Notice that the green bar on the parent strand lines up with the salmon bar on the newly synthesized strand. These bands are meant to highlight the complementary nature of the sequences of nucleotides of the opposing strands. They will also allow us to keep track of the relative lengths of the two strands as we go through the next two examples. So in the second box, we begin with one of the strands of a separated DNA molecule again. We begin the synthesis of a new complementary strand in the 5' to 3' direction. However, the DNA polymerase and part of the newly synthesized strand detach, all right? And what happens is they detach and slip backwards. All right. The DNA polymerase then reattaches the partially detached strand to a previous location on the parent strand that had already been copied. This detachment, all right, backwards slippage and then reattachment of the newly synthesized strand results in the creation of a small loop of single-stranded DNA that is not base paired with the parent strand. So that's what this looks like here. When the DNA polymerase reattaches to the parent strand, it continues to synthesize a new strand that complements the parent strand. However, when the strands of the new double-stranded DNA molecule are separated for another round of replication, the loop of the newly synthesized strand is straightened out. Because of the extra and repetitive synthesis of the newly synthesized strand, you can see that the salmon segment of the newly synthesized strand is longer than that of the green segment of the parent strand. As you can see from the depiction of normal DNA replication in the first box, the salmon and green bars should be the same length. Now, when the newly synthesized strand of the previous replication serves as a parent strand of this second replication, you can see that a brand new DNA molecule with a new inserted segment is created. This inserted segment is an insertion mutation. You can also see that the parent strand of the previous replication again goes on to serve as the parent strand in the second replication. Here you can see that both the green and salmon segments in this new DNA molecule are shorter than those in the extended molecule. At this point, you can see that we've already added a single strand from a separated DNA molecule to the third box, so we can now demonstrate forward slippage. DNA polymerase reattaches to the strand of DNA and initiates the synthesis of a new complementary strand in the 5' to 3' direction. Again, the DNA polymerase detaches from the parent strand, and a portion of the newly synthesized strand detaches from the parent strand as well. However, this time the parent strand slips backwards, and the DNA polymerase and newly synthesized strand reattaches farther up the parent strand than the location in which it detached. In this process, it is the parent strand that forms the loop of nucleotides that are not base paired with the opposite strand. While we can see that the parent strand has slipped backwards, much in the same way that the newly synthesized strand slipped backwards in the previous example, we can also think of the newly synthesized strand as having slipped forward. This is the rationale for the term forward slippage. Because of the loop of unpaired nucleotides in the parent strand, you can see that the straight green segment on the parent strand does not reach as far to the right as it normally would. In turn, the salmon segment of complementary nucleotides on the newly synthesized strand is also shorter. When the two strands of this new DNA molecule separate, for a second round of DNA replication, you can see that the newly synthesized strand from the first replication is now the parent strand. Because this strand was shortened from the forward slippage that occurred in the first replication, it serves as a template for a new shortened double-stranded DNA molecule. You can see that both the salmon and green segments of the DNA molecule are shorter than they would have been if normal replication had taken place. This deleted genetic material represents a deletion mutation. Again, you can also see that the parent strand of the previous replication goes on to serve as the parent strand in the second replication. Here you can see that both the salmon and green segments of this new DNA molecule are longer than those of the abbreviated DNA molecule. So what would cause the forward and backward slippage of the DNA polymerase? It turns out that many genes possess what are known as microsatellites and mini-satellites. Microsatellites are segments of DNA that have repetitive units of 2, 3, or 4 nucleotides. Because these units are arranged back to back, or in tandem, along the strands of the DNA, they are sometimes referred to as tandem repeats. Right? And then here we have a depiction here of these tandem repeats. Mini-satellites are analogous to microsatellites, except for the fact that each repetitive unit within a mini-satellite are variable in length, and those lengths can vary from 10 to 100 bases. Right. These microsatellites and mini-satellites create areas within the DNA molecule where misalignment of the parent and newly synthesized strand can easily occur if the DNA polymerase detaches and reattaches. In other words, because the two complementary strands repeat themselves so many times within a single region, it is not unlikely that the parent strand and newly synthesized strands will become misaligned when they are reattached after a temporary detachment. Whenever insertions or deletions occur as a result of misalignment within the microsatellite and mini-satellite regions during DNA replication, they are considered to be spontaneous mutations. Unstable repeat expansions are germline mutations that are thought to result from spontaneous insertions and within microsatellite regions. Genes with unstable repeat expansions possess microsatellite regions in which the repetitive units, usually 3 nucleotides, progressively expand in number from one generation to the next. The precise mechanisms of how or why these expansions occur within the germline are unknown. However, as previously indicated, the mechanism is thought to involve the backward slippage of the newly synthesized strand during DNA replication. As the repetitive units within the microsatellite units are progressively expanded from one generation to the next, the proper functioning of the effective gene and/or its product becomes progressively impaired. Several of your classmates have likely selected diseases for their application paper that are attributable to unstable repeat expansions. They will discuss these diseases in more detail at our final online synchronized session. And here we have a depiction of the Cag triplet expansions that occur in Huntington's disease. All right, and right here you can see that if you have the Cag triplets between 10 and 26, all right, then the person, okay, with this allele, particular allele with this number of expanded repeats is unaffected, all right, but they may then produce progeny, okay, which have an expanded set of repeats, okay. If you see that those people have 27 to 35 repeats in their allele, then they are still unaffected. However, if they have progeny, which also inherit the allele, albeit with an expanded set of repeats in the 36 to 39 bracket, then you actually start to see late onset of the disease. All right, and then finally, if that person goes on, okay, and passes that allele on to their progeny, all right, and there is a further expansion of the repeats to over 40, then you're going to get full penetrance and a progressively earlier onset of the disease. So far we've looked at spontaneous causes of insertions and deletions. However, insertions and deletions can be induced by mutagens as well. A class of mutagens called intercalating agents, and examples of intercalating agents would be acridine orange, ethidium bromide, and dioxin, become wage, or I'm sorry, the best way to say this is these intercalating agents, otherwise known, some examples would be acridine orange, ethidium bromide, and dioxin, become wedged among the base pairs of a DNA molecule. All right, so in this picture right here, you see acridine orange, all right, and then here you can see that it's wedging itself between the two strands, and in effect, it causes distortions of the shape of both of the strands. Okay, so the positioning of the intercalating agents distorts the shape of the affected strands. When the distorted strands serve as a parent strand in DNA replication, DNA polymerase is misled by the distortions created by the intercalating agents. As a result, the DNA polymerase ends up adding or subtracting one to a few nucleotides from the newly synthesized strand. As we demonstrated previously, the newly synthesized strand will then go on to serve as a parent strand in subsequent replication, and the mutation will be passed on. As we will show you next, if insertion and deletion occur within the coding sequences of genes, frameshift mutations can often occur. Frameshift mutations are especially troublesome as they can grossly change the amino acid sequence of proteins. All right, and this in turn will change the primary structure, which will in turn change all of the other hierarchical structures above that, and thus have significant impacts on the functioning of the protein. All right. So as a result, intercalating agents can cause significant functional disruption. So now let's go back and review frameshift mutations. All right. So the deletion or insertion of nucleotides within the coding sequence that are not multiples of 3 and or destroy part of a codon may result in the shift of the entire reading frame for that coding sequence. All right. This will change the identity of all the codons beyond that mutation. All right, and then a change in the identity of the codons may change the identity of the amino acids that they code for. So in this case, we're going to look at the start codon here in this exon of the beta globin gene. All right, and from here on end, we see the reading frame is established by these blue brackets, and you can see within each blue bracket the different codons that are distributed beyond that start codon. All right, and we're going to go along our way through the process of translation, okay, lining up the amino acids appropriately with their codons on the messenger RNA transcript. All right, but we now have a deletion mutation here at this cytosine. All right, as a result, we see a shift in the entire messenger RNA transcript, which leads to a shift in the entire reading frame. All right, so instead of the next codon coding for proline like it should, okay, to produce a normal protein, instead it codes for leucine. And then instead of the next amino acid coding for glutamic acid, instead it's coding for arginine. All right, and again, arginine. Instead of lysine, we get serine. Instead of serine, we get leucine, etc., etc., etc. And before you know it, we have something that looks nothing like beta globin once the different spliced out segments are fused together. Here in this final picture, just to kind of give you some perspective about how damaging these intercalating agents are, this is a picture of the Love Canal Superfund site in Niagara Falls, New York. And I encourage you to look at the wiki link that I gave um, that details this incident. And basically what happened is that in this area right here, a chemical factory, I believe it was Baker Chemicals, dumped an extraordinary number of drums of dioxin. Okay, and then they had covered it up with clay and dirt, and then they sold the land to a developer who then built a subdivision over it. Not only that, but they sold part of the land to the Niagara Falls School District, which built an elementary school on the site as well. And over the next 2 decades, they saw an alarming rate of leukemias and other cancers in the children who grew up here, and all kinds of other um, 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 mutagenic effects from these intercalating agents once they started to leak out from the drums. So this is in contrast, stark contrast from, for example, nitrous acid, which is only causing point mutations. All right, and you can see how, I have the example of how nitrous acid is derived from the sodium nitrite that we all are regularly exposed to in our processed meat products. Um, uh, uh, that is a relatively tolerable mutagen. All right, the damage is done instead of acutely, it's done over a long amount of time with significant exposure to the mutagen. Okay, and that is because our DNA repair mechanisms are successful in preventing those mutations from getting out of hand, and then also the mutations themselves are less damaging. But here you see with the intercalating agents, because these mutations are far more damaging in that they're more likely to cause frameshift mutations, the effects are much more acute and much more severe. All right, and that does it for our mutations lectures. Again, we're going to cover the chromosome mutations that are also classified as large insertions and deletions when we do our cytogenetics lecture. And I'll see you all then.